Hi, welcome to Unit 3 of Bio 193A, Metabolism and Biochemical Test. This is a chapter, or this is a unit that you would benefit, especially if you are taking the lecture course at the same time, to review the lecture material first. And if you are only enrolled in the lab course, you may want to review the OpenStax lecture book, which is free, because it's going to help your studying. So why do we use biochemical tests in the microbiology lab? They are very useful, especially in clinical microbiology, to characterize and identify bacteria. And in this particular lecture, and if you were doing a microbiology lab in person, you would be mostly doing these reactions one by one. But in microbiology labs, you will have this multi-test system when you can, test 15, 20 of these tests at the same time, and there are patterns that will give you codes and that will help your identification basically in less than 1 day. The biochemical tests are going to map to metabolic reactions, and I'm going to focus on 3 kinds: the carbohydrate metabolism, protein metabolism, and respiratory enzymes. And one nice feature of these biochemical tests that are very visual, so they're easy to observe, easy to decide is this positive or negative. So starting with carbohydrate metabolism. Again, you will benefit if you review the fermentation part, but most of these tests are going to focus on the ability of bacteria to ferment certain carbohydrates, particularly sugars. So phenol red broth, the name says it, so these are broth, and they contain a pH indicator called phenol red. And if you see in this slide in the middle, there is this red broth here, which is uninoculated. So the original, let's say, negative color is red. If a bacterium is able to ferment a sugar, then the color changes to yellow because the pH of the solution becomes more acidic, and that changes the color of phenol red. So what does a phenol red broth contain? It contains the phenol red, it contains sugar, and that would be one kind of sugar for each kind of broth. So you're going to have dextrose, which is the same as glucose broth, lactose, sucrose, and others. And it also contains peptone. And peptone is a protein digest, and there are bacteria that are unable to ferment. They are what we call obligate aerobes. An example of that would be, for example, Pseudomonas. Pseudomonas aeruginosa is a very common and very nasty bacterium, one example, but Pseudomonas is a very common obligate aerobe. Actually, they are not obligate aerobes, but they are non-fermenting microbes. So there are bacteria that will grow in phenol red broth because they are going to degrade the peptone. They are not going to touch the sugar. And in that case, the color actually will become a deeper kind of burgundy, deeper red, darker red, burgundy red color. So, but when the reaction is positive, which means that the bacterium is able to ferment, then you can observe 2 things. One is the change to yellow, which you clock as acid. And sometimes it also produces gas, and the gas is captured in this little tube. So this is a small tube that is inverted, so upside down. And if the bacterium produces gas, then the gas bubble gets trapped, and it's very easy to see in the Durham tube. So phenol red broth is usually reported as, or fermentation reactions are reported like acid gas, or negative, which would be a non-fermenter. Then there are also two tests in one. So the MRVP broth is a special broth that can be interpreted two ways. One, methyl red, that is the MR part, and Volksproskauer, which is the VP part. So among fermentation, there are many products, and some of those products are acidic, more acidic. This is tested by methyl red, and other products are more neutral, and that would be tested by the VP test. Bacteria that are, so MR and VP go basically the opposite ways. So if a bacterium is fermented, it's either MR positive and VP negative, or vice versa. Simmons Citrate means so this is a slant, and it has this very nice green color, and what it measures is the capacity of the bacterium to use citrate as its only carbon source. And again, if it's positive, if it can use the citrate, then the positive reaction is blue. There are also tests that measure the ability of the microbe to break down macromolecules. Now remember that bacteria are very small, so usually they don't, and they have cell walls around. So it would be very hard for bacteria to, let's say, bring in a large protein or a large polysaccharide inside the cell to degrade it. So some bacteria are able to produce what we call exoenzymes, which break down whatever is around them. So if you look to the left, that is gelatinase assay. So it's an enzyme that breaks down gelatin, and again, cooking. So you use gelatin to make something more solid. If an enzyme has gelatinase, then the gelatin will melt. So your gelatin cube, which as you can see, it's solid, it will become liquid. Casein looks at the ability of breaking down this protein called casein. It's a milk protein, and it is used in an agar that contains milk, and positive reaction is observed with this clear halos around the colonies. And finally, you can also look at the degradation of lipids. So on the right-hand side, you have the methylene blue tributyrene agar. So this is an oil, and again, a positive agent. As you can see in the upper panel, it will be able to degrade that lipid, which means that you will have a clearing around it. Now, why is this important, or why is this indicative? If you think of a bacterium that is inside the human body, and we are going to talk about that when we talk about pathogenicity, a bacterium, if it needs to invade, needs to penetrate the human body, the human tissues. So bacteria that are able to break down proteins, polysaccharides, and so on, they tend to be nastier, because that means that they can advance faster. They can break down, like, let's say, connective tissue, and that way they can invade human tissues. And if, cellulitis is it's the medical word for inflammation of tissues, and very often it's caused by bacteria that are advancing in the tissue, breaking down proteins, polysaccharides, and so on as they advance. Some more large molecules. So starch hydrolysis looks at the production of an enzyme called amylase, which breaks down starch. Urease is a protein that breaks down urea. So you may remember that urea is a nitrogenated compound that is a way for proteins to break down. And urease is tested by a urea broth. And if you have a positive reaction, you are going to see this change to a very nice kind of fuchsia pinkish color. And then you have a number of other tests that will look at degradation of amino acids. This slide shows what we call the IMViC reactions, and this is like a panel that is very often used, and in this particular set, these are all gram-negative bacteria that are often present in the GI tract. So IMViC stands for indole, MR, VP, and citrate test, and again, it's very commonly used for gram-negative rods called enterics, because they cause enteric infection. And you can see that if you do all those reactions, you are it's easy to figure out which is which because they don't have the same pattern. I don't think there is, like maybe Enterobacter, Serratia. So some of them are very similar, but then you can use some additional tests in order to differentiate between two, but at least it will give you an idea of who is who. Then you have reactions related to respiratory processes. So oxidase is a shorthand for cytochrome oxidase C, and it's just an enzyme that can or cannot, may or may not be present in the respiratory chain. And there are a lot of ways how you test that. Sometimes you just take a little strip, like an oxidase strip, take a little bit of the bacterial suspension, and if there is a positive reaction, you will see that kind of dark purple color. Sometimes you just have a liquid reagent that you drop on top of your culture. And catalase is an enzyme that breaks down hydrogen peroxide to oxygen and water. If you have ever cut yourself and you put some hydrogen peroxide on the wound, you see bubbling, that is catalase for you. It's a very useful enzyme to have in the presence of oxidative agents. So in the diagram here, or the picture here, shows a catalase test done on a slide. So you have a glass slide, you take a little bit of your bacterial culture, you smear it on the slide, and you add a drop of hydrogen peroxide. If you have bubbles, it's positive. If it's negative, if there are no bubbles, it's negative. So it's very useful for two. Let me see if it's in the next slide now. So when you are looking at Staphylococcus versus Streptococcus, so they are both cocci, so they are round. They are different in how they look and their arrangements, but one very simple way to know if it's staph or strep is with catalase. So Staphylococci are catalase positive, and Streptococci are catalase negative. And the same is applicable to bacilli and Clostridium. It is kind of simple in the sense that Clostridia, by definition, are obligate anaerobes. They do not have catalase, and bacilli are always catalase positive. So when you do your Labster simulation of the week, you will be seeing many of these reactions. So hope that you review this before doing the simulation. Thank you.